Hello guys, today I'm going to be doing a bio past paper, okay, for the board AQA, Science A Unit Biology B1. So if this video hits 1 million subscribers, sorry, it's not subscribers, sorry, it's if it hits 1 million views, I would be happy, right? Yes, it would be. So let's start. We'll go with biology, unit biology B1 with the code BL1HP, starting with higher tier. Right, so what they are asking us to use is black ink. It's a black ballpoint pen, so you have to use black ink, okay? The total marks for the paper is 60. Right, let's start with the first question. Many people in the UK take sleeping pills. The drug thalidomide was developed as a sleeping pill in the 1950s. In the 1960s, how do you pronounce this? Thalidomide, I think, was banned. Recently, thalidomide has been used to treat other diseases. Name one disease thalidomide is used to treat now. Mainly content learning. Biology is mainly content learning. So basically, at the end of the day, learn your stuff, right? So it's going to be called leprosy, okay? So table 1 shows information about the development of a new sleeping pill. Okay, great. So now what is the shortest time taken to develop a new sleeping pill? So the shortest time. Let's see, we can see the time taken in years, right? Let's see what's the shortest time. Okay, should be, what is the shortest time taken to develop a new sleeping pill? Right, so we have to understand time taken for test and trial in years is 1 to 4, 2 to 4, it's about shortest time in phase 3, it's about like 6, maximum 6, 2 to 3. Okay, it's 6, so plus 4 plus 2 is 6, right? So time taken or trial in years is 2 to 4. That is technically it's 6. So you can see a comparison there. So it's 6. What is the range for the number of volunteers needed to complete all the clinical trials for the new sleeping pill, right? What's the range? Complete all the clinical. So we have to understand this, right? The range that should be from, let's say from 1,120 to 5,600. So now why am I giving this range, right? Number of volunteers, right? So we have to look at the number of volunteers. That is about greater. So the number of volunteers is about 100 to 500 patients, right? And we have about 1,000 to 5,000 patients. So the range of volunteers that are needed to be completing all the clinical tasks is that we add them up to get us 5,000 plus 500 plus 100, 5,600, with 1,000 plus 100 plus 20 is 1,020. So how I calculated this is for 1,000 plus 100 plus 20, and for 5,600 it's 5,000 plus 500 plus 100. This is how I get the values for the range. Alright. Drugs are trialed to check for side effects on people. Give one reason why drugs are trialed. Now this is mainly, okay, why drugs are trialed. Because there's a test for toxicity, because it's poisonous, right? Because drugs are poisonous. Drugs are poisonous, right? Figure 1 shows the impact on the health of the population caused by drugs from different sources, right? Legal non-prescribed drugs have a greater impact on the health of the population than illegal drugs. Suggest 2 reasons why. So our first reason should be understanding this pie chart. This is a pie chart, so it shows us the impact on the health of the population caused by drugs. Okay, as we can see, a major chunk goes for the legal non-prescribed drugs. As you can see, it's a dark shade, like a medium shade color. So this has a greater impact rather than illegal drugs. Now, the main reason is because, right, because more people take the legal drugs, unlike the illegal, right? Take the legal drugs. Take the legal, non-prescribed drugs, right? They take the legal, non-prescribed drugs. And these drugs are more readily available because they are legal, right? These non-prescribed drugs, which are legal, are more readily available. Right? Alright, so now drugs change chemical processes in a person's body. Why is it difficult for a person to stop taking certain drugs? Okay, it's because one answer, it's addiction. It's one answer, it's addiction. It cannot change because it's one mark, it's addiction, because they won't stop due to the dopamine intake in their lives. Right. Name process X. Okay, figure 2 shows the mass of carbon added to and removed from the atmosphere each year. So we know that if it's plants and algae, most of it, photosynthesis, is a process which adds carbon back to the atmosphere. So I know directly it's going to be called photosynthesis. Photosynthesis is a process that directly adds carbon back to the atmosphere. Calculate the mass of carbon added to the atmosphere by respiration per year. So this is something that we have to understand. Okay? So the mass of carbon added, calculate the total mass of carbon added. So by looking at the scale, I know that there's a difference of 50 divided by 10 squares, right? So if you do 50 divided by 10 squares, that's 5 billion tons of carbon per block. I think it's per block. Right, so we know that this is 50, alright, and then 50 plus 5, this is 60, this is 50, this alright is 5 times 5, 25, plus 5 is 30, processed by algae. Actually, sorry guys, photosynthesis is not a process that adds carbon to the atmosphere. It's not actually photosynthesis, because I just looked at the diagram again. It's actually saying removed from the atmosphere. So you have to remember 3 things: that photosynthesis is a process where carbon dioxide is removed from the atmosphere by plants and algae. It's removed from the atmosphere. So carbon is taken by the plants for the process of photosynthesis. In the process of photosynthesis, it is respiration and combustion which causes carbon to be added back to the atmosphere. Okay, sorry guys for that. So to calculate the mass of carbon that's released, so this is 10. So we have 50 plus 60 plus 30 plus 10. That should give us about 150 billion tons of carbon being added back to the atmosphere, right? So that's 150 billion tons carbon being added back to the atmosphere. That is pretty significant. So, right? That is pretty significant. 150 billion tons. Now, if there's a way to correct this, you guys can correct it. I mean, it doesn't matter. It's all right. Okay. But by checking the marking scheme, I've seen that it's about like they're showing about like 140. I think it's 150. So now, how scientists are concerned that the mass of carbon in the atmosphere is changing? How does the data in figure 2 support this data? So right, we have to analyze that. We have to analyze the portion where the carbon is added and as well as removed. So by finding out the difference, right? So we can find out the difference by calculating the number, the total of this block. So that's 100 plus 5 times, more like that's 5 per block. So you're technically having, okay, this is 100. And what is this section here? We can see that there's a 5, so we know it's 5 times 5. It's 125 plus 5 times 1, 2, 3. That's 140. So 140 is being removed from the atmosphere, right? So we know that 150 minus 140, that's about 10 billion more tons that are being more added than removed, right? 10 billion tons that are added then removed from the atmosphere. So we can see that there's an increase of 10 billion tons being added to the atmosphere rather than removed from the atmosphere. All right, let's move on to the next question. Okay, so you guys have commented, I think some of the viewers have said that my handwriting has to change. All right, so I'll write better next time. Okay, so gardeners sometimes use weed killers. Okay, get straight to the point. What do we have to analyze? We have a table, make the investigation fair, give one variable the gardener controlled in the investigation. So he wants to get rid of daisy plants, use weed killers. The gardener must record the number of daisy plants, make sure the solution of the weed killer each had a different concentration. That itself is a highlighter that it's an independent variable. So basically we have put 5 dm, okay, concentration. So we have to use the same volume solution used. So what we can say is we can use the same volume of each solution used. Same volume of each solution used of about 5 centimeter cubed. All right. So the gardener decided that the result for a concentration of 20 arbitrary units of weed killer was anomalous. So this is why the gardener decided this result was anomalous. Okay, so for a concentration of 20, anomalous values are those that do not meet up. So we can understand there are more daisies growing, right? The number of daisy plants 2 weeks after using weed killers, we can see that their concentration of weed killers after 2 weeks, they're growing more for 20, right? So decided that the result for 20 a weed killer was anomalous. So by adding 20 weed killers, right, of arbitrary units, the number of daisy plants have grown. So there are like increased daisy plants, you can mention that for mark. There are increased daisies growing after use of weed killer, right? That's pretty cool. And understanding that it does not fit with a pattern like the others, which should be decreasing. This should be decreasing right after using weed killer. Right. So why did the gardener put 0 arbitrary units of weed killer in one area of land? So this could be used as a control factor, mainly as a control factor. That's what I'm going to say, is a control factor, or it could be used to compare it to other areas. As a control factor, that could be one, or he wants to like check other factors that are not affecting the results. That in this case is the daisies. Now the gardener concluded that the best concentration of weed killer to use all over the lawn is 100 arbitraries. So just why the gardener cannot be sure about this conclusion. So the gardener cannot say that 100 is the best, because if you look 2 weeks after using the weed killer, right, is 0. But do you see one other trend is that while using 80 concentration of weed killer, it's also 0. 2 weeks after, the number of daisies have also been decreased. So basically you have to mention that 80 arbitrary units of weed killer, right, kill the daisies after 2 weeks as well. Killed all the daisies after 2 weeks. killed all the daisies after 2 weeks as same as the concentration of 100 arbitrary units, right? All right. Okay, next one. So in this question, you will be assessed on using good English, organizing information clearly, and using specialized terms where appropriate. Okay, let's get straight to the facts. Is that in your answer, you should refer to the role of plant hormones. Do not refer to the use of artificial use of plant hormones by gardeners. So you have to understand what plant hormones are and answer this question indirectly. So there are some marks awarded for the quality of your answer, and if you want to achieve those 5 to 6 marks with a level 3, you need to refer to at least like one environmental factor that plants respond to, or at least one associated response. That is, you have to give like a reference to a named hormone in order to score those 5 to 6 marks. So I'm gonna just list down like some pointers that you can use, right? Okay, so like how plants respond to different environmental factors. You can say that there's a change in environmental factors such as intensity of light, and change in concentration, change in the moisture or water levels, change in the moisture and water levels around the plant, All right. Okay, describe how the environmental factors can affect growth and the direction, right? So if there is if there is more light, right? So more light will lead to, you can say more light will lead to the shoots growing upwards and growing towards the light. Okay, but the force of gravity can cause, the force of gravity causes the roots moving downwards towards the moisture and water levels. Okay, all right. Okay, now this can be mainly because of the hormone. This can be mainly, so remember, named hormone, mainly because of the hormone auxin in the plants, right? And the unequal distribution of hormone causes unequal growth rates. All right. Okay, you can also name other hormones if you like. You can say the higher concentration of hormone causes faster growth rate in shoots. Higher concentration of hormone causes slower growth rates in roots as well. Okay, so let's move on to 4A. You have to remember, this is key terminology again. What is a pathogen? We know that pathogens are disease-causing microorganisms. So you can just call that disease-causing microorganisms. Okay, microorganisms. Alright, so we know that a pathogen are disease-causing microorganisms. It could be either bacteria, virus, or fungus that causes infectious disease. So bacteria may become resistant to antibiotics. How can doctors reduce the number of bacteria that become resistant to antibiotics, right? So reduce the use of antibiotics, right? So we have to reduce the use of antibiotics, reduce the use of antibiotics, and reduce the use use of antibiotics. Why do I say antibodies? Antibiotics for mild infections. Right? So we can also say we can use a wide variety of antibiotics, but you can understand that. So scientists grow microorganisms in industrial conditions at higher temperature than those used in school laboratories. Which temperature would be the most suitable for growing bacteria in industrial conditions? Remember that the temperatures should not be too high because it can denature these enzymes, right? And it should not be too low, so it should be like the perfect medium rate of temperature. So in this case, it is 40 degrees. So now what is the advantage of using the temperature? Just like I said, it could denature the enzymes if the temperature is 100 degrees Celsius. So we need the perfect temperature. So you can say that the microorganisms can work faster without being denatured, right? Natured in high temperatures. So that means they don't get burned or destroyed due to high temperatures, so that these enzymes or these microorganisms don't get destroyed. Now 5a, organisms compete with each other. This is more like competitions, and most of the time two seaweeds are covered with water. So I like these kind of questions because it's related more with analysis questions, analyzing the idea. So bladder wrack has bladders filled with air. These are two types of seaweeds. Okay, we know the bladder wrack grows more quickly than saw wrack. Now suggest an explanation why. So okay, so what are the things we need to know? These are filled with air, right? Most of the time the two seaweeds, they live in similar seashore habits. At the same time, they're covered with water, right? Now bladder wrack grows more quickly than saw wrack. Now the reason is we can look at the adaptations, right? So it's filled with air. So because it has air, it can get more light because there's more numbers, because maybe the bladders are floating when the tide is there. So basically this air just gives an assumption that they need more light or they need oxygen to survive. So you can say that the bladder wrack, it gets more light, right? So photosynthesis occurs faster. So photosynthesis occurs faster because the tides are flowing, because the bladders aid floating because they're filled with air, right? Or you could just mention that there's more biomass glucose starch produced. I mean, that's also fine. So now anglerfish live at depths of over 1,000 meters in clear water. Sunlight doesn't usually reach more than 100 meters deep. Many anglerfish have a transparent lure containing a high concentration. You would have seen in many movies these fish appear to be in the movies, and they seem to attract the protagonist into it lures the prey into its mouth, I think, when it wants to eat, but the ball of light mainly acts as a distraction. Okay, so they have a transparent lure light containing a high concentration of bioluminescent bacteria. Bioluminescent bacteria produce light. So just an advantage of the anglerfish of having a lure containing bioluminescent bacteria. Like I said, it lets the anglerfish attract its prey. Okay, so it can attract prey because it acts like a distraction. So that itself gives you an understanding that the anglerfish, you can say for 2 marks, sorry guys, the anglerfish will be able to attract prey to get food, right? Because it's hungry to get food. All right. This can also be used as a sign to help the anglerfish. The light, right? Helps the anglerfish. This light produced by these bioluminescent bacteria helps anglerfish to see in the dark, right? Fish to see maybe the prey or its predators, right? Or its predators in the dark, in depths as it lives in the dark as it lives in depths of over 1,000 meters. Now this is pretty interesting. You can say that this light helps the anglerfish to attract prey, as well as to help it see the prey and the predators, because it's living at depths of over 1,000 meters. So anyone who's living in depths of over 1,000 meters is going to be really dark. And also you can say that the anglerfish will be able to attract mates to reproduce, sorry guys, and it lets the anglerfish to see predators without being eaten. So it acts like a safety mechanism for it to survive. Now, in January 2011, more than 600,000 people collected results for the UK National Bird Survey. Okay, so people recorded the number of each species of bird they saw in 1 hour on one day in their garden. Now, some of the results are shown in Table 3. So you can say that a student looked at Table 3 and said, in the UK, we have about house sparrows are more common than what of blackbirds. Now, suggest 3 reasons why the student's statement may not be true. For 3 marks, we have to identify and compare those values that are in Table 3. Okay, so what can we make the assumption of? Right, house sparrows are more common, because you can see the mean number of birds seen is higher. Okay, that's more common. Okay, that's percentage of gardens in which birds are seen. Okay, it's pretty lower. Are they giving any more values for us? Okay, there's nothing else. Okay, so just why the student's statement may not be true, because you can see the percentage of gardens in these birds were seen is pretty less. Okay, and whereas we have a higher chance of seeing other birds, because blackbirds are more seen in more gardens, right? So because blackbirds, just by looking at the table, you can see that blackbirds are seen in higher percentages in higher gardens, right? In higher percentages in more gardens, right? So this is only done in gardens. Maybe the change in locations could affect the results. Change in locations and time it was done, timeline and time it was done in is not a fair conclusion. Does not give it a fair conclusion. Does not give it a fair statement. Does not give it a fair statement of conclusion. Statement of conclusion, right? That's what we can understand. And sometimes human error can be caused as well. Human error can be caused when people miscalculate number of birds seen. Calculate number of birds seen. Alright, number of birds seen. Alright. So, a survey in 2012 was done the same way as the 2011 survey. Figure 5 shows us changes in the percentages of gardens in which some birds were seen from 2011 to 2012. Calculate the percentage of gardens in which goldfinches were seen in 2012. Use information from Figure 5 and Table 3. Okay, we have to calculate the percentage of gardens. Okay, we can see the percentages of gardens in the Y scaling, where goldfinches were seen in 2012. So by looking up here, we can see goldfinches, right? And by looking here as well, we can see goldfinches as well here in the bar chart, and it comes to about 2012. That's a change in particular, right? So change of percentage gardens, the birds are seen, 2012. Okay, survey done 2011 shows the changes from 2011 to goldfinch. change in percentage, we can see the percentage of goldfinch were 33.5. So this is coming up to 80. So what we should be doing is we should be analyzing the percentage change gardens where the birds have been seen. So how we're going to solve this question is a statement that we have to ask ourselves is that the total for goldfinches, total percentage, calculate the percentage of gardens in which the goldfinches were seen. Change in percentage of garden, okay, is 80. So it's a positive increase of 80%. So we know we can do is 80%, 80 divided by 100, right? So we can do 100 plus 80 first. Let's do 100 plus 80. That should give us 180, right? So 180 divided by 100, that's about 1.8 increase. So 1.8 increase, the number of goldfinches seen here is 33.5. So what we're going to do is, because this is a change, we're just going to go with increase. So because there's 1.8 increase, 1.8 multiplied by the value that is stored in table 3 for the goldfinches, and the percentage is 33.5. So 1.8 times 33.5 is equal to indirectly 60.3. That's a percentage of gardens ever in which goldfinches were seen, 60.3. Okay, so just two reasons why goldfinches were seen in more gardens in 2012 than in 2011. This could be maybe the change in temperature due to global warming. Change in temperature due to global warming. There you go, that's one mark. Due to global warming. Due to global warming, and there could be fewer predators, right? And then maybe less disease in 2012 and fewer predators. This could have resulted in more finches being seen, goldfinches being seen. The question is about the nervous system. Okay, the human nervous system is a pretty interesting chapter. Describe the function of receptors in the skin. Receptors are those that detect changes in the surroundings. Receptors, they detect changes in the surroundings. They detect changes in the surroundings, and they convert information. They convert this information to impulse, causes movement as well, right? So this sends the impulse to the sensory neurons, that is the brain in this case. It could be the brain, but you guys get the idea. Right, response is caused when information in the nervous system, it reaches an effector. Now there are two types of effectors. Complete table 4 to show the different types of effectors, the response each effector makes. So there is one called the muscle effector, that's one you need to remember, and one called gland effector. Remember, there's 4 marks, so they're split into 1 mark, 1 mark, 1 mark, 1 mark, okay? 4 marks eventually. Muscle helps to contract, it helps in contraction itself, and whereas gland muscle, it helps to secrete and produce chemicals. It helps to secrete chemicals and produce chemicals. All right. Some effectors help to control body temperature. Give one reason why it's important to control body temperature. This could be to prevent maintaining body temperatures. Like I said in the previous question, if you have high temperatures, right, the enzymes won't work effectively. So maintaining temperatures, right, controlling body temperatures will help the enzymes work at their best conditions, will help the enzymes work best, and prevent damage to these enzymes, and prevent damage to the enzymes, prevent damage, right, to the enzymes without being destroyed by overheating of temperatures. This, like I said, so basically by increasing the temperature, you will indirectly denature these enzymes. So by preventing enzymes from being denatured by the temperature increase is pretty vital. Next question is about what is a gene. Remember, a gene, there's a certain some call it a section of DNA, but you have to say a section of DNA that codes for a particular characteristic or codes for a specific protein. I've seen that in most marketing schemes. So in this case, we're just going to call it a part of a chromosome that controls a characteristic. You can also say it's a piece or a section, or I'm just writing here, a section of DNA that codes for a specific protein. It codes for a specific protein. So now this definition is really precise. So you have to score those 2 marks by saying part of a chromosome or controls a characteristic, or say it's a section of DNA or a specific protein. Right. Okay. So scientists have developed a way to change specialized cells back to embryonic-like cells. Okay, iPS can be changed. Okay, that's great. Scientists plan to use skin cells from endangered species of monkey called a drill, and change these cells into iPS, and can be changed into an egg cells or sperm cells. Oh wow, after fertilization, the embryo can be inserted into the womb of a female of a non-endangered species called a mandrill, and the mandrill is closely related to the drill. So this is basically done for the adult cell cloning chapter. So describe similarities and differences between the iPS method and adult cell cloning. So you can say that both iPS and cell cloning, adult cell cloning, they use body cells. They use skin and body cells. Both transfer, right, embryos into the womb, right? And the difference is that iPS uses sexual reproduction, whereas adult cell cloning does not involve this. And whereas adult cell cloning does not involve this, adult cell cloning does not, and you can say that iPS has no nucleus transfer, and offspring is basically kind of like genetically different. Offspring is genetically different from the parent. Okay, let's move on to the next question. So just one advantage of trying to preserve endangered species such as drill for maintaining the ecosystem or maintaining the food chain balance, right? Maintaining the food chain and the ecosystem and the ecosystem, right? So the idea of retaining biodiversity or maybe economically useful in the future, but it's mainly the idea of maintaining food chain and the ecosystem. What is meant by balanced diet? So a balanced diet is a diet consisting of the right amounts of nutrients and right amounts of energy. Okay. All right. Cholesterol. Name the organ in the body which produces estrogen. It's basically ovaries. Okay, ovary. 1 mark. What effect does estrogen have on the reproductive cycle? Okay. it helps to inhibits, this is mainly content learning, inhibits maturation of the eggs, right? Maturation of the eggs, and causes like stimulates some hormone, I think, most probably. Now, estrogen is a naturally occurring steroid hormone. Give one artificial use of a steroid hormone in the body. So this can be used, like they said, in the body. You can use to stimulate muscle cell or muscle growth in the cells. So it can be used in oral contraceptives as well. Right. So now, figure 6 shows the effect of the mass of cholesterol in the diet. And you have to give the idea, there's a graph showing us the cholesterol levels, and describe the effect of increasing the mass of cholesterol in the diet on the mass of cholesterol produced. The green marks include full data. So what you have to understand is that the mass of cholesterol produced by the liver and produced by the liver is this dotted line. This is the liver, and this is the blood cholesterol levels. Right, there's a small rate of decrease and then a bigger rate. So there's a small rate of decrease, and then bigger rate of decrease, right? So there's about a difference, about like maybe it touches at 90, so it changes, the change of rate, there's a change of rate touching at 900 milligrams per day. Okay, so that's one answer. Okay, now which part of the liver is responsible for controlling the production of reductase? It's mainly the genes. Okay, this is part of the cholesterol in the diet, right? So that's mainly genes. Now, high blood cholesterol concentrations increase the likelihood of heart and circulatory. Doctors can prescribe statins to control the concentration of cholesterol. Explain how statins work. So statins are these kind of like these. They reduce the production. So it reduces the production of cholesterol by liver. Or you can mention that it helps to reduce the reductase production and absorption, reduces the absorption of cholesterol by the intestine, and statins help to destroy the cholesterol by breaking it down. So this seems to be the end of the questions for this paper. I hope so you liked this video. Please do like, share, and subscribe with your friends or family or anybody who you guys keep in touch with. And if you want me to do more past papers on specific subjects such as computers or physics or math, please do comment below. I would really appreciate it. Otherwise, I think that this is the end. Not the end. I mean, there's more videos to come. Well, this is it. I think that's it. All right. Bye, guys.